Good morning, good afternoon, and good evening to all of you who have joined in the second edition of the Genomic Variant Analysis and Interpretation course. I am Dr. Vinod Skaria, a scientist with the CSIR Institute of Genomics and Integrative Biology, and with my colleague Dr. Shahida Moosa, we invite you all to this inaugural session. This is the second session or the second edition of the Genomic Variant Analysis and Interpretation course, and we're happy to note that we have over 1,500 odd participants to this course from 40 odd countries, making it one of the largest programs educating clinicians on interpretation of the pathogenicity of genetic variants. In this inaugural session, we have a stellar list of speakers who kindly agreed to share their experiences on how they've used genomics in the clinics and how they have moved forward in using model systems to understand the biology of the disease and also discover newer therapeutics for many of the rare genetic conditions. And I, with my colleague, look forward to a wonderful session ahead and having you all for the next 16 weeks over this course. Thank you very much. It's my pleasure to introduce Dr. Anurag Agrawal for the inaugural speech for the Genomic Variant Analysis and Interpretation course. Dr. Anurag Agrawal is the director of the CSIR Institute of Genomics and Integrative Biology. After his undergraduate medical studies at the All India Institute of Medical Sciences, he trained in internal medicine, pulmonary diseases, and critical care at the Baylor College of Medicine, Houston, USA, followed by a PhD in physiology from the Delhi University. After serving as an assistant professor at Baylor, he joined IGIB in 2007, eventually becoming the director in 2017. His interests are in this part deployment of emerging technologies towards healthcare, and is a co-chair of the Lancet and Financial Times Global Commission on Governance Health Futures 2020. He has an illustrious career and received many awards, including the Shanti Swarup Bhatnagar Prize in Medical Sciences in 2014, the National Biosciences Award in 2015, and elected fellowships of the Indian National Academy of Sciences and the Indian Academy of Sciences in 2018. He's also a senior fellow of the Wellcome Trust DBT India Alliance for Clinical and Public Health Research and on advisory groups of the WHO. The floor is all yours, Dr. Anurag.
It's my pleasure to speak to you all today about genomics, genomic technologies, and the future of healthcare. I'm going to start with this very simple slide. A slide on a drug called Zolgensma, which, if given to a patient with spinal muscular atrophy in time, within the first initial diagnosis time, early in life, can completely prevent the development of neurological disability and death. The question then for us clinicians is, how can we ensure that people born with this disease in India are able to not progress to such disability given the technology exists? So what are the challenges? The first challenge is timely diagnosis of such disorders. It could be via simple test, it could be via NGS. That does not matter. The point is that tests exist and should be made accessible to people. And number two, given that this is $2 million for one injection, how do we make it available to people without only asking for charity? This brings up to a very important point about the health economy and our vision for what we see as emerging technologies' role in the global health economy and India's role within it. Healthcare is the largest services sector worldwide. It is catching up in India, of course. It has the highest rate of growth. The domestic value may reach about $500 billion US. The necessary key drivers are present in India as in the rest of the globe. Aging populations, new technologies that are more effective, growing insurance penetration, and a growing commitment towards universal healthcare. Even if you look at it from the science and technology perspective, of 6 trends recently talked about in Nature Medicine, 3: big biomedicine, read, edit, write genomes, and personalized medicine are heavily dependent upon genomics. if you were to take big biomedicine, you'd be imagining the system from grounds up, from knowing the blueprint of the body to how the organs and the systems function, and trying to predict from the organization and the blueprint what are the ways in which problems can occur. You could also go the other way around. You could start with techniques like next generation sequencing or genomic diagnostic. You could take things that you know how they affect health. You could apply them at scale of individuals to public. You could try to either correct them at the level of the blueprint itself, genetic correction, gene editing, CRISPR-based technologies, or you could try to use drug-based therapies to optimally in such people based on what is known to be the best line of treatment for similar people elsewhere. In sickle cell disease, we've already seen full editing-based corrections, bone marrow transplants, and a normal life for those people since they've been observed. At animal level, we have even seen another extreme. We have seen the idea of introducing beneficial mutations into normal genes that can have long-term beneficial effects, even though there was no genetic defect per se to start with. And at a level of only pure experiment in labs, we've even seen creation of bacteria with recoded synthetic genomes. Now, these have tremendous applications, as I will discuss as I go forward. On the left, you see two Nobel laureates, Charpentier and Doudna, who won the Nobel for the CRISPR therapeutics. And often people talk about how, given a defective gene, you can correct it. But on the right, I have another person, Helen Hobbs, who found that within normal genes, there can exist some people who have a beneficial mutation. In a gene called PCSK9, there is a natural extreme lipid-lowering variation that is actually a mutation that makes PCSK9 non-functional, that lowers your cholesterol tremendously. This lowered cholesterol can also be achieved by drugs targeting PCSK9 or antibodies targeting PCSK9. And so some of you are probably already using such drugs in your patients who cannot take statins. In monkeys, it has been seen that if you target this normal gene by CRISPR technology, or even by blocking the protein in other ways, you can lower the cholesterol for years by a single shot. This offers the ability of tuning human genes and human genomes for maximum benefit. But this is not something that clinicians should take lightly. Today, we are talking about somatic cell mutations, where even if we were to treat some people, it will not be passed on to the next generation. So at least the risks will be confined to a single generation. But tomorrow, when the technology is perfected, where all would you apply it? Would you do it for skin color? Would you do it for hair length? Would you do it for muscle strength? These are controversial. Would you do it for longevity? I guess you might. Would you do it for germlines? Would you do it in such a way that the benefits of this therapy would be passed on to offspring, thereby creating a set of people who could be said, in one sense of the word, to be better? It's a very tough, very difficult, ethically debatable point as of today. Only somatic gene editing is allowed, but we as clinicians need to think about it and have a voice on such topics in the future. I think India is a wonderful place for genomic research. A large population, a diverse genetic pool, representing all the populations across the world, a melting pot of the world, so as to speak, historically, a stratified population with multiple small endogamous groups. And while one should never talk about genetic disease being a benefit, but the close-knit family structure and communities in India make India a wonderful place to do discovery and develop therapies for genetic diseases. IGIB has spent many years working on both angles, using high-end science for discovery through programs like Guardian that I'm sure you will hear about, as well as delivery, in which you create affordable, accessible diagnostics for people through a program called GoMed, which again I'm sure you will hear about. This circle must continuously be evaluated and completed in India, because only investing in high value discovery, which is very expensive, is not sufficient to meet the needs of people. And that is one reason why I believe that clinicians who keep the ultimate goal in mind are very important people to participate in genome science and imagine its future applications for the world. The way I assess the current situation is there is demand locally for existing technologies, especially if we can make it affordable. There is even a growth of the upper middle class with capacity to pay for higher value, lower certainty wellness products. There are growing insurance measures so that low cost, high value measures can be really scaled to the billion scale. There is a recognition of global need for new technologies in this space, particularly for things like gene therapy and genetically engineered cell-based therapies, where I think India has a major role to play by indigenization, lowering costs, as well as complete new imagination. So if I were to end this talk with a few key area messages, I think we need to invest with clinicians and researchers working together for locally applicable precision wellness services, not just precision sickness, precision wellness. We should be imagining the optimum genetic screening and testing for Indians and NRIs. We should be imagining the way in which to best communicate, maintain, and transfer such results. And you hope Vinod will speak to you later about the genome card. We should be thinking about ways in which we can use other aspects of genomics, not just the human, like microbiomes, to give people wellness advice. And we should be talking and developing precision therapeutics, including gene therapy and genome engineering. With that, I thank you all very much. Thank you.
Greetings, everyone. It's my pleasure and honor to introduce Dr. Lisa Schimmenti, who is at the Mayo Clinic, USA. Dr. Schimmenti is a professor of pediatrics and co-director of the Medical Scientist Training Program, popularly known as the MD-PhD program. She is also a consultant at the Department of Clinical Genomics, Biochemistry, and Molecular Biology. Dr. Schimmenti's research focuses on genetic conditions associated with hearing and vision loss. Dr. Schimmenti conducts clinical research studies in zebrafish to model human conditions. Her focus areas has been genetics of hearing and deafness. She also models human diseases in zebrafish. She actively investigates families and children that are affected by rare and undiagnosed genetic conditions using a number of methods, including careful clinical observation, documentation, along with sophisticated genomics and functional techniques that could provide answers to long diagnostic odysseys. Dr. Schimmenti is a member of the Center for Individualized Medicine and the Department of Clinical Genomics, and where they have pioneered the diagnosis of patients with rare and undiagnosed diseases. In summary, Dr. Schimmenti's research aims to improve patient care through identification of precision medicine-based novel therapies for hearing loss and to develop improved algorithms to reach a diagnosis in individuals with undiagnosed and rare diseases. Of special mention to this particular meeting and all of you is her expert. She has served as an expert member in ACMG and AMP variant interpretation guidelines, which have been widely published and circulated. She has also been an expert member in defining standards for clinical exome sequencing in a number of disease conditions. With this, I would like to invite Dr. Lisa Schimmenti to deliver her keynote lecture to all of us on the topic entitled Genomic Diagnosis in Rare and Undiagnosed Diseases. Dr. Schimmenti, it's my pleasure to invite you to deliver the talk. Thank you.
Hi, my name is Lisa Schimmenti, and I am a clinical geneticist at the Mayo Clinic in Rochester, Minnesota. And so I am really pleased to be here today to talk to you, and deeply honored by this invitation by Dr. Sivasubu and Dr. Scaria. And so the topic of this morning's talk is genomic diagnosis in rare and undiagnosed disease. And this is part of the genomic variant analysis and clinical interpretation course. The objectives for today's talk is to understand the definition of a rare disease, describe the impact of making a genetic diagnosis in a critically ill child or newborn, to describe the benefits and limitations of whole exome sequencing and whole genome sequencing, and to understand some of the genetic counseling issues regarding whole exome sequencing for families who receive results. So why are we here? First of all, I really commend all of you who are taking this course, because it really speaks to your commitment to the care of individuals with rare and undiagnosed disease. It also speaks to your commitment to precision medicine, that there is a specific diagnosis for individuals who may not have a diagnosis, and that we will find the correct and precise diagnosis to manage that individual. And then to your educators and to your mentors and leaders of this course is to really celebrate their commitment to training the next generation of investigators who can move effectively and efficiently into the care and diagnosis of individuals with rare and undiagnosed disease. So how do we define rare disease? And there are a number of working definitions that are available. The first is the Rare Disease Terminology Working Group that defined a rare disease as 40 to 50 cases per 100,000 individuals worldwide. The European Union definition is very similar. It's a condition affecting fewer than 50 per 100,000 individuals. In the United States, the definition is a condition that affects fewer than 200,000 citizens or individuals within the United States. So for many of you, this is a review, but I want to make sure we're all working and starting together. So we're working on the genome, and that's our set of genetic instructions for how the body forms. There are over 3 billion base pairs of nucleic acid in our genome, and this nucleic acid is packaged into chromosomes, and we have 23 in a haploid set, but our diploid set, 1/2 from each parent, is 46 chromosomes. Each genome contains about 22,000 genes, and that's what we call the exome. And this is only 1 to 2% of the genome. And each of us is nearly the same as everyone else on this planet. I just want to quickly run through some genetic terminology that I'll be using throughout this talk that I hope will make things easier moving forward. So an allele is an alternative version of a gene. So if there's a DNA sequence difference between one version of the gene or another, we'll call that an allele. Sometimes you may see that abbreviated as big A or little a, as different versions of the same gene. A genotype refers to the genetic constitution of an individual. And so remember, we have 2 of every gene, one from our mom and one from our dad. And so we could have 2 big A alleles, a heterozygous gene, which is a big A and a little a, or little a in that, in terms of which alleles a person has. But a phenotype is the expression of a genotype. So the physical or clinical trait that an individual has is their phenotype. A homozygous individual or a homozygote is somebody who has 2 alleles that are identical. A heterozygote is someone who has 2 alleles that are different. When we talk about dominant, we're talking about the phenotype. That means the appearance of an individual. And that appearance is based on having 1 copy of the gene that has a genetic variant that's medically actionable or pathogenic, or what we used to call a mutation. A recessive phenotype is only observed when both alleles, one from each parent have a mutation or a variant. A carrier is an unaffected individual who is heterozygous for an allele with a pathogenic variant, but does not have any phenotype. And variable expression is a difference in severity or phenotype in individuals with the same genetic variant or same genotype. So a genetic variant refers to a difference in DNA sequence between your patient and what we refer to as a standard consensus DNA sequence. There are three types of variants that we talk about with patients. The first is benign, meaning that there's a sequence difference, but there's evidence to support that that sequence difference does not confer any disease and is not medically actionable. A pathogenic variant or a likely pathogenic variant are what we formerly called a mutation. And these are variants in DNA sequence that do confer a disease or a phenotype and are medically actionable. And then if a variant does not meet criteria for being benign or does not meet criteria for being pathogenic, it is called a variant of unknown or uncertain significance. And those may not be medically actionable, but further research is needed to try to determine if that variant is truly benign or pathogenic. So let's walk through two cases of individuals that benefited from exome sequencing to solve a medical mystery. And so the first case is a 2-day-old full-term baby girl who appeared normal, so she was non-dysmorphic, and she was transferred into the neonatal intensive care unit, or NICU. At birth, the baby cried, but then had trouble breathing, and so required positive pressure ventilation and improved. She had good scores at delivery, APGAR scores of 7, 8, 9, and her prenatal ultrasounds were normal, and the amniotic fluid around the baby was normal. But at 24 hours of age, she became cyanotic and developed difficulty breathing. This is her family history. And so squares are men, circles are women. In this family history, this is the mother, this is the father, and this is the baby at 8 days of age. This baby had pulmonary hypertension diagnosed by the neonatal intensive care unit staff. This baby's mother also had another child with a previous partner that passed away at 48 hours of age and had pulmonary hypertension. This baby has a brother through her father with another partner who's 6 years old and healthy. Going through the mother's family side, there is a first cousin who is 9 years old, who at birth also had pulmonary hypertension, and another first cousin through the mother's brother who had pulmonary hypertension in the newborn period. And the mother had a sibling that passed away at 6 months of age due to sudden infant death syndrome. The mother herself had a history of asthma and chronic obstructive pulmonary disease and had a normal echocardiogram of her heart. Whole exome sequencing was performed on this baby. And whole exome sequencing is the simultaneous sequencing of the entire coding region of the genome, so that 1 to 2% of the genome. And this comprises approximately 22,000 genes. This testing works best if we have both parents available. And when we evaluate this sequencing, we can look for recessive conditions, dominant conditions, new genetic changes, which we call de novo conditions, and that are conditions that are based on a gene list that we generate based on the patient's phenotype. When we do this testing, we can get an answer about 30% of the time in neonatal cases. And this is from a small series at our institution. With the first 20 cases, 7 cases were solved. And we can do this testing quite rapidly in less than 10 days. And there are some centers in the United States that can now do this in 48 hours. When we talk to parents before doing genetic testing, we discuss a number of issues with them because it's important that families understand what type of testing is being performed and what that testing means. Some of the things that we could identify through exome sequencing includes non-paternity, meaning that the person that's believed to be the father may not truly be that individual. We could also identify that parents are related to one another, so consanguinity. We could identify carrier status. That means an individual may be a carrier of a pathogenic variant in a gene that's unrelated to their condition, but we find out that they are carriers, and that information is important for families as they make future reproductive decisions. As I mentioned, the yield is 30%. I think everybody in this field got very excited when we could finally sequence exomes and now genomes, and we thought we would solve every case, but the yield right now is still around 30%. There are also a number of findings that we call secondary findings. So when we consent families to exome sequencing, we talk about conditions that we're not really looking for, that are there to solve the child's condition, but are important for families. And these include genes that are important for medical care, but they're really not the reason we're doing testing. And so we give the families the option to know about these conditions and the option to opt in or opt out to know this knowledge. And these include genes for cardiomyopathies or aortic aneurysms, cancer syndromes like the breast cancer gene, BRCA1 and 2, and then also genes that are important for the way we deal with anesthetic. And so a condition called malignant hyperthermia is genetically mediated, and genes for malignant hyperthermia are also part of the panel of secondary findings. There are limitations to variant interpretation, and this particularly is important for families that are underrepresented in genomic databases. And this represents a health disparity worldwide. We use normal variation in a database called NOMAD. And this database is from the Massachusetts Institute of Technology. And this database is highly sampled for people who are of European heritage and fairly well sampled for people who are of East Asian heritage, but really has poor representation of the peoples from Africa, the Middle East, South Asia, Latin America, and indigenous people of North America and other continents. And so there's ongoing needs to improve representation of genomic variants in all groups. So what were the results on this baby that we identified? So 9 days later, exome sequencing revealed that there was a pathogenic variant in the gene TBX4. And this gene is associated with autosomal dominant pulmonary hypertension. And so this is the result of the actual result from that baby. So in the first column is the name of the gene. Under it is the transcript number of that gene. This is the variant. This is the C period is the cDNA at position 702 in the cDNA at the +1 position rather than a G. There is now an A. This is the genomic location, and this is the condition associated with this genomic variant, and that this is inherited as a dominant. And this child is heterozygous. This variant was inherited from the the child's mother, and the classification of the variant as pathogenic. And so there's a description of this variant. And so this variant is associated with a condition called autosomal dominant pulmonary arterial hypertension and ischio-coxo-patella syndrome. And so individuals can have small patellas on their knees, as well as having pulmonary hypertension. And so this was really important for the team to understand why this child had pulmonary issues and helped in this baby's management, as well as help this family understand why they had a number of children who passed away. So here's a second case. This is a 24-year-old mother who is pregnant for the first time. Ultrasound at 22 weeks of her pregnancy identified brain malformations, specifically cavum septum pellucidum and agenesis of the corpus callosum. This child had wide space eyes or hypertelorism, and the orbits were asymmetric. And then extra digits on the hand and foot were noted, so polydactyly. An intrauterine MRI was performed at 27 and 5/7 weeks, confirming hypertelorism and cerebellar hypoplasia, in addition to the ultrasound findings at 22 weeks. Genetic testing was performed prenatally. A chromosome microarray was performed, and this is a test to look at all of the chromosomes and identify whether any regions of chromosomes are deleted or duplicated. And this child had 46 chromosomes and 2 X chromosomes, so a girl. They also did fluorescent in situ hybridization, and this was normal. So they were looking for any increased chromosome number for chromosome 13, 18, and 21. And so exome sequencing was performed, and the laboratory identified a pathogenic variant in the gene GLI3. We can call this GLI3. There was a duplication at position 4,431, resulting in an early stop codon in the coding region at codon 1478. This variant was not found in either parent, so is de novo. GLI3 is associated with 2 different genetic conditions, a condition called Grieg cephalopolysydactyly syndrome that's associated with having extra digits as well as brain hamartomas. There's also another condition that is allelic to Grieg cephalopolysydactyly syndrome called Pallister-Hall syndrome. Labor was induced for this family at 33 and 1/7 weeks because the mother developed hypertension. So she had preeclampsia and she was quite severe. The baby weighed 2.88 kilos, which is the 95th percentile, and was 51 centimeters long. And at delivery, this baby was limp, did not cry spontaneously, and was resuscitated. The APGAR scores were worrisome at 1 and 5 minutes, but became better by 7 minutes, so 2, 5, and 7. And the baby had apneic spells, and an electroencephalogram was performed and was normal. So there was no evidence of seizures for these apneic spells. These are pictures of the baby. The head circumference measured 33 centimeters. The eyes were widely spaced. There was postaxial polydactyly that you could see here from both hands, and then both feet also had polydactyly. On day 12 of life, this baby began to have an enlarging head size and was having more apneic spells. And an MRI was performed and showed hydrocephalus and ventricular enlargement. This child had also other abnormalities, including punctate periventricular blood hemorrhages, had a small posterior fossa and hypoplastic inferior vermis and enlarged fourth ventricle. And so it was the knowledge of this child's genetic condition that really helped the team to move more quickly to do an MRI and then consider CSF diversion using a VP shunt. So all in all, the solve rate for whole exome sequencing, at least at our institution, is out of 970 cases, 23% were solved, 4% were likely solved, and another 2% were partially solved, for an unsolved rate of 71%. And this is from 2012 to 2019. These numbers are improving. What's next? We're really moving to genome sequencing, and this is the entire 3 billion base pairs. This testing has improved capabilities, including the ability to identify deletions and duplications in the genome, as well as the ability to detect triplet repeat disorders. The coverage over whole exome sequencing is also improved because there's all of the genome is being sequenced, not just the exomes. So there's actually a step called the capture step in an exome, and there's variability in this capture step. If there's no capture step and everything is sequenced, this provides better coverage. Our knowledge base for interpretation is constantly improving. And so our ability to interpret non-coding parts of the genome is also improving. And as the cost comes down, we may be able to replace other testing like microarrays. We're also getting to the point where we're looking at RNA sequencing. And this is really useful to detect a condition called mosaicism, where an individual does not harbor a genetic variant in every cell of their body, but in some of the cells of their body. And this can be missed by genome sequencing. So RNA sequencing is an improvement to detect low-level mosaicism. It may also be useful to detect genomic abnormalities that are not picked up by genome sequencing because of the bioinformatic approaches that we use. And those bioinformatic approaches may miss a gene fusion event caused by 2 chromosomes fusing together as the result of a translocation. And so RNA sequencing has been really helpful. Also, some variants may lead to abnormal splicing during the development of messenger RNA. And so RNA sequencing can help determine if splice variants are pathogenic. And so we're starting to use this more, particularly in cancer genetic diagnosis. We're also looking at proteomics and metabolomics, and we're starting to look at abnormalities in protein production that result from abnormal genomic sequencing. And there's a lot of progress being made in post-translational modification of proteins, such as conditions known as congenital disorders of glycosylation. So as a practitioner who takes care of children who have undiagnosed diseases, you may want to engage the genetic service earlier in the course of care to help manage patients, and consider exome and genome approaches that might be performed rapidly and improve the medical management of children who are critically ill, or provide clinical insight when their status changes. I want to thank my colleagues at the Mayo Clinic who provided slides and information to prepare this talk, particularly members of the Department of Clinical Genomics and the Department of Laboratory Medicine. And I also want to thank my colleagues at the Center for Individualized Medicine who work with us to solve undiagnosed cases. And so thank you very much for allowing me to participate in this course.
Hello, everyone. I am Vamsi Yanamandra, Physician Scientist Faculty at the CSIR Institute of Genomics and Integrative Biology, New Delhi, India. It is my pleasure to introduce our next speaker, Professor John McGrath from King's College London, where he is currently serving as a chair for Department of Dermatology. Over the past 3 decades, Professor McGrath has pioneered the discovery of many novel genes involved in the pathogenesis of genetic skin diseases, setting up the tone for genomics research in these diseases. In addition, he has also pioneered the discovery and development of innovative therapeutic solutions to alleviate the sufferings of these patients. It is a journey I always cherish to listen to, and I hope the listeners will have a treat listening to the same. I hereby invite Professor McGrath to deliver his keynote lecture to the audience. Thank you.
Yes, hello everybody. This is John McGrath from London, and it's my pleasure to be joining you today to share with you a story about translational research for patients with inherited skin diseases. It's my journey, it's national, it's international, it's a journey of fun and discovery, and I hope very much that you'll enjoy the presentation too. So yes, I'm coming from here in London, Guy's Hospital, cloudy skies, but a wonderful place to work. Let's get on with the lecture. So here you are. Here are patients with inherited skin diseases, also known as genodermatoses. And the translational research activity is twofold. It's all about diagnosis, and then ultimately, of course, it's about treatment. So what about diagnosis? Well, this is what we really want to try and diagnose. Babies like this with something really challenging. And of course, the journey that's happened is that making an accurate diagnosis and offering better clinical management and treatments is what we want to do as dermatologists, as physicians. In this case, the answer to this particular mystery was an autosomal recessive sterilopathy. And there is a treatment for this. Straight to treatment with cholesterol and statin because of the disruption of lipid metabolism. For years, of course, we would have wondered how can we make such an accurate diagnosis? And of course, now it's really very much the product of next generation sequencing. But it wasn't always like that. For me, this has been my clinical research activity for many years, the gene hunter. What do you think? Indiana genes? Maybe this is taking it too far. But this has been my ambition, to try and identify genes through global collaborations through many friends in different parts of the world. And it's been quite successful. Here is a new autosomal recessive disease caused by inflammation, scaling, pustules, and infection. And this turns out to be homozygous loss of function mutation in epidermal growth factor receptor. Fascinating new insights into EGFR signaling. And here's one. Dermatologists will be familiar with dermatofibromas, these firm fibrous lumps, but the familial forms thereof seem to be caused by variants in factor 13A antigen. And that gives us new insights into fibroproliferative pathways and maybe ways we can modify scarring and wound healing in the future. And indeed, I mentioned that I have had this global collaboration, and indeed over the last 30 years, my lab has been involved in the discovery of all these different genes and diseases, some new and some really just trying to assign genes to particular established diseases. What a fantastic experience it's been. To try and discover the molecular basis of patients like this, the two main techniques that we've used in the last century were candidate gene analysis and genetic linkage. So this was before next generation sequencing was ever dreamt of. So typically, a next generation sequencing free world involved candidate gene approach. So for a child like this, what we would do is look for pathology. As dermatologists, we take skin biopsies, and here we can see that the mid part of the epidermis seems to be a little bit loose, what dermatologists call acantholysis. We can focus in on those cell junctions and see that the desmosome junctions are abnormal. That tells us, let's go looking at the desmosome proteins. And indeed, the patient on the left has an absence of one particular desmosomal protein, which turns out to be something called plakophilin 1. We can then do the Sanger sequencing, identify the homozygous nonsense mutation, and we have a diagnosis. So this was the world's first genetic disorder of desmosome junctions found by a candidate gene approach, great for autosomal recessive diseases with loss of function mutations. On the other hand, for large pedigrees like this, with an autosomal dominant condition, the old technique was to get large pedigrees, to do microsatellite markers, to do fine mapping, and eventually try and map to a particular chromosomal locus, and then sequence laboriously all the genes within that particular interval. And in this case, for this family with autosomal dominant telangiectasia oropharyngeal cancer syndrome, it turns out to be the DNA repair gene ATR, so we can give it a name and publish. But of course, next generation sequencing has been with us now for about 13 years, and Indiana Genes has evolved to make the most of the technology. So thinking of human beings and sequencing extensively and filtering through literature, through algorithms, through processes, and identifying pathological mutations has been our modus operandi. So if we reflect on what has been the impact of next generation sequencing on gene discovery and clinical diagnostics, it's that more than 170 new skin disease gene associations have been established, and that includes more than 35 brand new genodermatoses. So on average, that's 3 new diseases per year. Here are some of the conditions that we have worked on with a combination of different gene discovery techniques over the last 30 years. And it's been predominantly to do with blistering diseases. So here you can see mutations in collagen 17, plakophilin 1, dystonin, and exophilin 5 causing skin fragility. And here is the most recent one that we worked on, autosomal recessive hypotrichosis skin fragility syndrome. Turns out to be another desmosomal disease, but this time it's biallelic loss of function mutations in desmocollin 3. So epidermolysis bullosa is the name given to this group of inherited blistering skin conditions. As a clinical scientist, once you discover the genes, you have an obligation to try and establish laboratory diagnostics as part of a service. And that service will also extend to the clinical world as well, so you need to provide better treatments for patients. So here is our diagnostic laboratory as set up after those initial gene discoveries. You've got to think about what you're going to do. So in the case of EB, were we continuing to do skin microscopy? Was it all going to be DNA sequencing? Were we going to do cell work? Were we going to make provision for various omics technologies? In terms of the sequencing, how much of it were we going to do in-house and how much was going to be outsourced? These are all the considerations you have in setting up a diagnostic laboratory as part of the translational research process. Anyway, here is our lab as it is currently flourishing. And of course, those clinical services for patients with epidermolysis bullosa are complex. It's not just dermatology. There are these multidisciplinary care aspects that one needs to coordinate as part of your translational research activity as an academic clinician. But of course then, epidermolysis bullosa is just one of many inherited skin diseases with huge unmet need. There are more than 1,000 different genetic diseases affecting the skin. And so that means we need more activity. We need national and international initiatives for the diagnosis and treatment of patients like this. And in the UK, one of those national initiatives has been the 100,000 Genomes projects, recently completed with all of those genomes having been sequenced. So the plan here was to do something at a national level, to sequence whole genomes from National Health Service patients, focusing on rare diseases, including EB, and some common cancers and some infectious diseases. And more than just sequencing, the genome data was linked to medical and health records, and the data was shared appropriately. And that meant that this purpose of trying to improve knowledge of the causes of disease and the treatment of those diseases and the general care of patients with those diseases was very much improved. It's still work in progress, but certainly a great national initiative. Here is one example of the 100,000 Genome Project in the UK. So two families, British Pakistani, British Indian, not known to be related to each other, one family known to be consanguineous, the other not, but 4 affected individuals. And they shared a particular clinical phenotype. You can see dyspigmentation. For the dermatologist, you'll recognize poikiloderma, which is a combination of hyperpigmentation, hypopigmentation, telangiectasia, and atrophy. There were also some hair abnormalities, minor eye abnormalities, and some keratoses on the soles. Skin biopsy confirmed poikiloderma. So they were recruited to this 100,000 Genomes Project. You have to specify particular gene panels to look at, and that was done. However, no potentially pathogenic variants were identified, so you can take it to a data analytical stage beyond that with a gene agnostic approach. And doing that, this particular variant popped out, a gene I'd not heard of before. And this was present in the affected individuals from both families. Patients 1, 2, and 3 were all homozygous. Their parents were heterozygous. Patient 4 was also homozygous. And because we have whole genome data, we're also able to establish that there was a block of homozygosity in both families, in keeping with a common ancestral allele. LTV1 stands for low temperature viability protein 1, and this is part of the ribosomal complex. We know that the dysfunction is in the 40S subunit. More functional work to be carried out on that in the future. But this turns out to be a brand new autosomal recessive genodermatosis, and we're giving it the name LIFAC syndrome, which stands for LTV1 associated inflammatory poikiloderma with hair abnormalities and aqueous keratoses. We'll see how many other cases of that exist. If you want to read more about it, it's in press in Human Molecular Genetics. So what about patients like this with these different genodermatoses? Beyond diagnosis, my real thoughts in clinic are what can I really do to help patients like this, particularly patients with epidermolysis bullosa or the severe forms thereof. So for epidermolysis bullosa, you can see the physical burden of disease. You can see the challenge in trying to reduce blistering, improve wound healing, and so on. Sometimes we get lucky. Have a look at this case here. You've got a before and after case. You can see widespread crusting and skin fragility, and then somehow we've come up with a treatment. Have we come up with a treatment for EB? Unfortunately not. What was the treatment here? Ivermectin. Ivermectin for EB? Some of you are already there. This turned out to be epidermolysis bullosa simplex with scabies. So it's very important to be a good clinician, even, and remember your dermatology when you're managing patients with rare diseases. And here is a patient from Iran with a mild form of recessive dystrophic EB. You can see the crusts and erosions and so on. And then there's an after picture. And we've given the treatment zinc. Is zinc a treatment for dystrophic EB? No. This individual from a consanguineous family also happened to have a second autosomal recessive disease, acrodermatitis enteropathica. So the zinc is treating that. So here you have the importance of accurate diagnosis and sequencing data, and then linked to a treatment that you can prescribe for patients. If we stick with the treatment of epidermolysis bullosa for a while, it's very clear that wherever you are in the world, there are very limited treatments available. Popping blisters, giving various dressings, supporting nutrition, and so on. Now, we all need mentors, and my mentor in the field of epidermolysis bullosa was the late Robin Eadie, who pioneered many developments in this field. But I still remember his final ward round before he retired. And he said to me afterwards, you know, I've looked after this patient called Claudia for 20 years, more than 20 years, but sometimes I do wonder whether I've actually done anything useful for her at all. And that's always a reminder for us as clinicians that tomorrow is too late for many patients. We need to keep pressing on. So these are babies with EB, and some of the work that Robin Eadie did was to help characterize the basic pathology. And so understanding the dermal epidermal junction in skin and employing young researchers like this to study the basic pathology. That's where we were 30 years ago. And we were able to establish that EB was a collection of monogenic skin fragility diseases with defined molecular pathology. So far, so good. And it seemed like a linear journey, just to go pathology, diagnosis, treatment, everything would be fixed relatively quickly. Not the case. Of course, we have, like many other research groups, tried to focus on gene therapy and cell therapy, but successes are few and far between. The biggest success in gene therapy in dermatology is this one, reported in Nature just about 5 years ago. The gentleman in the bottom right is Michele De Luca, and he did ex vivo gene therapy, putting in one of the laminin genes, LAMB3, into keratinocyte holoclone stem cells and doing ex vivo grafting of 80% of this particular child with junctional EB skin. A great success, but it's just one patient. Lots of other early phase clinical trials of gene therapy in EB, targeting keratinocytes and fibroblasts and skin equivalents, putting back the defective gene into those cells and grafting them, injecting them, applying them to the patients, but very much early phase. One of the most recent exciting applications of gene therapy in EB is this one, using a herpes simplex vector. So the company Crystal Biotech have just recently reported a phase 3 trial, a double-blind placebo-controlled trial looking at paired wounds in the same patient. This is a gel, like a Vaseline ointment, in which the herpes simplex vector is carrying type 7 collagen gene, and it's being applied to wounds. And the data at 6 months are pretty impressive. Extended use trials are now underway. But it doesn't have to be high-tech and expensive all the time. Gentamicin, an aminoglycoside antibiotic, is also a form of gene therapy in that it can read through transcripts which have got nonsense mutations in them to improve mRNA stability and make some protein. Here you can see type 7 collagen being expressed in skin basement membrane zone. There are issues, of course, in terms of sensitivity and antibiotic resistance, but this is now being tried not only topically, but intradermally and intravenously. We await the results in patients. And another phenomenon that we need to be aware of is revertant mosaicism. Sometimes there is natural gene therapy going on in the skin of these patients. You can see these patches where one of the recessive mutant alleles has rebooted and recovered. And we can exploit that phenomenon. Grafting of revertant keratinocytes or punch grafting of those skin from those revertant patches can be used for translational benefit. This is great work. And now we can also see that commercially cultured epithelial autografts can be made from this revertant mosaic skin. And for that success, we have to thank another individual, the late Marcel Jonkman, a mentor of Dr. Vamsey, who discovered revertant mosaicism and helped drive this translational journey to help patients. We hope there's more in store from this particular phenomenon before too long. The latest technology, of course, that we're aware of is gene editing, particularly base editing and prime editing. So base editing is where you can correct a single nucleotide, and prime editing is the sort of form of gene editing that will be useful for small indels and so on. Here is an example of gene editing, base editing from our own lab. You can see on the left a heterozygous nonsense mutation, the C and the T. And then using this particular base editor from David Liu's lab in Harvard, you can see we can get 100% correction. We dearly look forward to taking this forward in trying to develop new treatments for patients with this and other mutations. It's exciting, but there's a long way to go. In terms of gene therapy and cell therapy, we are indeed on a journey. For cell therapy, one of the big global advances for patients with inflammatory blistering diseases has been the use of intravenous allogeneic mesenchymal stromal cells, you can see the improvement in wound healing and the lessening of inflammation in this particular individual. We know that these mesenchymal stromal cells from whatever source release a soup of anti-inflammatory immunomodulatory cytokines and growth factors that help improve the patient's skin, wound healing, itch and pain, and so on. So what about patients like this? We do need to do other things as well, and we can't always rely on gene therapy and cell therapy. What about some easy fixes with readily available cheap medicines? Well, look at this Taiwanese teenager here with skin dyspigmentation. Now, as dermatologists, we biopsy and we see melanophages and melanin, but it doesn't get us closer to what's going on. We can look at the pedigree, and that doesn't really tell us very much. This could be if it's genetic, autosomal recessive, or de novo dominant. So we turn to whole exome sequencing, and we discover autosomal recessive mutations in a gene called ABCD4. Finding those mutations gives us a diagnosis. It's called cobalamin J. It's to do with vitamin B12. There's no way, as clinicians, we'd have jumped from that clinical appearance to a vitamin B12 pathology. But we can now give a treatment, B12, and within a relatively short time, you can see the dyspigmentation improves. So this is a good, quick, cheap fix. Here is another good fix for patients, not quite so cheap, but mechanistic dermatology in practice. This is a mutilating keratoderma, Olmsted syndrome. We know there are gain-of-function mutations. It's autosomal dominant in a vanilloid receptor called TRPB3. We can then do biology tests to work out why does this hyperkeratosis occur. Turns out to be TRPB3 induced activation of that gene I told you about before, epidermal growth factor receptor. So rather than giving retinoids, which is what dermatologists would normally do, maybe we could use low-dose EGFR inhibitor to target those same pathways. And when that's done, you can see the dramatic clinical improvements. Mechanism-based therapy, a fix for patients, targeted therapy, translational research success. We've also gone on to start wondering whether biologics can help in treating genodermatoses. After all, they're wonderful for psoriasis and eczema and many other conditions. Well, autosomal recessive congenital ichthyosis, which we know as a genetically heterogeneous group of diseases, hard to think you can have a monotherapy for these patients. But work done by Amy Paller's group in Chicago has shown that many of these individuals have Th17 abnormalities. And so repurposing some of those anti-IL-17 therapies may prove useful for patients with ichthyosis. And the early clinical trial data suggests that drugs like secukinumab may be helpful for about a third of patients with inherited forms of ichthyosis. More data to follow. And here's a patient with a desmosomal genodermatosis called SAM syndrome. It's desmoglein 1. The patients get a severe dermatitis, but the treatment here is not to target desmoglein 1 directly, but to look at the skin. And you can see there's a huge amount of interleukin 17 in lesional skin in that superficial dermis. And that's why secukinumab turns out to be a great treatment for patients like this. It's not the only biologic that's useful for our patients. Ustekinumab has also shown clinical impact, so targeting different interleukins. You can see here a form of autosomal recessive ichthyosis with mutations in NIPAL4 responding to ustekinumab. And here, patients with erythrokeratoderma and mutations in desmoplakin responding to ustekinumab. And patients with pityriasis rubra pilaris resulting from variants in CARD14 responding dramatically to ustekinumab. And more anecdotal reports. Here is the calcium pump abnormality, the autosomal dominant disease, Haley-Haley disease. This responds to dupilumab, which targets Th2 cytokines IL-4, IL-13. This is normally a treatment for atopic dermatitis, but it helps anecdotally patients with Haley-Haley disease. And dupilumab can also help some forms of itchy EB. You can see these very lichenoid lesions that respond again to targeting Th2 cytokines in this single patient. And even JAK inhibitors like oral tofacitinib, JAK1, JAK2 inhibition, can dramatically reduce the itch and improve the well-being of patients with dystrophic EB. Clinical trials are needed, but anecdotally, we have a window of observation that maybe this is one way forward to improve patients' diseases. But those are all very expensive, and we need to get back to the cheap and cheerful. Now, it doesn't come much more cheap and cheerful than statins. And drug library screening by colleagues in the University of Dundee have shown that this can be useful in patients with pachyonychia congenita, or at least some of them. So if you have a mutation in keratin 6A, then a statin, a simple statin, can be extremely helpful for patients. And there are clinical examples of that being the case. So understanding lipid biology is very useful. So here you can see the statins, HMG-CoA reductase, and that's pachyonychia congenita. Well, just downstream from that is the mevalonate pathway, and we know that this is relevant to dermatology as well, because several different forms of porokeratosis have abnormalities in this part of the pathway. There are mutations that are reported, and this upsets cholesterol metabolism, and there is a buildup of various upstream metabolites. But we can target that. We can do something to alter those mevalonate pathway metabolites simply just by giving a topical statin. And so here is a patient with a form of porokeratosis, disseminated superficial actinic porokeratosis, DSAP, twice daily 2% lovastatin ointment for 6 weeks, and you can see the lesions are cleared. Mechanism-based therapy, cheap and cheerful. And of course, if you look at that lower part of the pathway where cholesterol is upset, there are lots of other genodermatoses like Child syndrome and Conradi-Huneman-Hapley syndrome that have abnormal skin and other features because of this cholesterol pathway abnormality. And we can treat those, or at least the skin part of those conditions, using topical statin and cholesterol combinations. It's very helpful for patients. Mechanism-based therapy. But what about repurposing drugs to help patients with genodermatoses? Apremilast, it's very expensive, but it's out there for psoriatic arthritis and other indications. We know how it works in terms of targeting various inflammatory cytokines. Well, it's now been repurposed to help some patients with oral ulceration in Behçet's disease because of that commonality in IL-10, Th17 and NF-kappa B signaling pathologies. So this is drug repurposing. And what about patients with dystrophic epidermolysis bullosa? Well, we've carried out gene expression studies looking at wounded skin in patients with recessive dystrophic EB and the intact skin. We've carried out various heat maps, gene analyses, and we've asked the question, could there be any drugs that could induce similar changes to promote wound healing? So when you pursue the bioinformatics and see what the computer programs predict, it tells us that a good theoretical treatment for wound healing in dystrophic EB could be methotrexate. How cheap and cheerful is that? And probably this should spark an international collaboration, and in resource-poor countries, we should be thinking about a clinical trial of methotrexate in this form of EB. I think it will improve wound healing. And my final story for you, just to round off this lecture, is a new therapy for EB, perhaps, and maybe a whole lot more, which gives context to this translational research activity. Again, mentors of mine and colleagues. In the center, Professor Yoni Uito, and on the left-hand side, my great friend and colleague Katsuto Tomai from Japan. We saw that patients with recessive dystrophic EB who underwent bone marrow transplantation did very well. The skin improved, and there was new type 7 collagen in some, but we didn't know how it worked. We didn't know which bit of bone marrow. We didn't know which cells from bone marrow were coming out. Where did they go to into the skin? Where did they particularly finish up? And what did they become? We knew we couldn't go on with bone marrow transplantation because it's too toxic. We needed better understanding of what goes on. Turns out those key cells are a subpopulation of mesenchymal stem cells, and what's driving the blue arrows here is a particular peptide called high mobility group box 1, HMGB1. And so I moved to Japan to try and understand this particular pathology. And this Nobel Prize winning mouse was the reason for going. So this mouse, if you make a wound on that mouse's skin and then put a skin graft on it, you will create a sort of hypoxic bioreactor and induce crosstalk between the GFP cells in the bone marrow and that bit of skin. It needs to survive, and those cells will go out and make the skin survive. A skin distress signal, SOS, save our skin. And there is a biopsy, and you can see that those green cells do result in dermal infiltration, but also after several weeks, you can still see cells in the epidermis. And so, to cut a long story short, what happens is that in wound healing and in blistering, those hypoxic keratinocytes in the blister roof release the usual suspects: hypoxia-inducible factor 1 alpha, stromal-derived factor 1 alpha. But the new kid on the block is HMGB1. And this is amplified by TNF alpha on wound healing macrophages. It enters the circulation. It then drives forward the mobilization of a subset of bone marrow cells, mesenchymal stem cells, through this particular CXCR4 chemokine receptor. They're recruited to those hypoxic areas of damaged skin, and they then transdifferentiate into new keratinocytes, and skin is restored. Lots of science to back up that particular claim. And so HMGB1, what Katsutotomai has done is to make a recombinant peptide to the key part of the protein, that A box, HMGB1 fragment, and then started to give it intravenously to release those MSCs and to boost wound healing. In Japan, it's already been completed a clinical trial in moderate severity EB, and the results are promising, even better than intravenous MSC cell therapy. But it's not only the skin. It's also been tested in a cardiac function model of myocardial infarction in rats, and most intriguingly, in human clinical trials in stroke, the data are now being assembled for publication. If we can really show that this is a treatment for stroke, then there is a blockbuster drug on the horizon. And there are multiple indications in dermatology, from EB to venous leg ulcers to tissue hypoxia and beyond. And of course, for me, hopefully when COVID allows, plenty more reasons to return to Japan. So if I summarize where I think we've got to in this translational research story, we've certainly got better at diagnostics over the last 30 years. That can't be denied. But in terms of the translational journey, we have only seen a limited number of high-tech gene and cell therapy trials. I think we really need to have much more focus on mechanism-based therapies targeting inflammation, and to keep this global collaboration going so that we can benefit and improve the lives of patients with EB and indeed all genodermatoses and other genetic disorders. So that's my takeaway. I think maybe we can help a bit, and soon perhaps we can do quite a little bit more. I hope you've enjoyed listening to this talk. If you want to contact me, do email me, and hopefully I'm around for the Q&A as well. But thank you very much.
It's my pleasure to introduce the next speaker, Professor Shahida Moosa from Stellenbosch University in South Africa. Professor Shahida Moosa is an internationally trained clinician scientist. She's a medical genomicist who combines the knowledge and skills of clinical genomics, bioinformatics, molecular biology, and molecular genomics to lead the program on rare genetic diseases in South Africa. After completing her specialty training from Wits University, she has obtained her PhD from the University of Cologne in Germany, followed by a brief postdoctoral stint at the Harvard Medical School in Boston. Professor Moosa is very passionate about bringing genomics to Africa and using the technologies to benefit African patients, especially those who are still remaining underdiagnosed or undiagnosed. She's committed to mentoring younger scientists and to skills transfer and capacity building in Africa. She also has an illustrious career and has also served as editorial board member in a number of journals, including Clinical Genetics, the European Journal of Human Genetics, and she has been an expert in 2 ClinGen expert panels, the ClinGen syndromic disorder gene expert panel, as well as the ClinGen skeletal disorders gene curation expert panel. Today, we would be delighted to hear her experiences in using genomics in Africa. The floor is all yours, Professor Moosa.
Hello everyone, I'm Shahida Moosa and I'm a medical geneticist in Cape Town, South Africa. Thank you very much for inviting me to address you today. Genomic variant analysis and clinical interpretation are very important skills to master, and I wish you very well for a stimulating course which will definitely equip you. I, as a clinician scientist, along with my clinical duties in the hospital, I also run a lab with graduate students, and we do variant analysis and interpretation every day, both in the clinic and in the research lab. Today, I'd like to give you a little insight into the mission I'm working towards to benefit African patients with rare diseases. The talk is entitled, Dare to Think Rare, an African Perspective. As a medical geneticist, my patients can be of any age group, from unborn babies through to adults. For the majority, they find themselves at some point or the other on a unique journey. For some, their journey begins during pregnancy, when the doctor finds that there is something not quite right. For others, the joy of holding their newborn in their arms is overshadowed by whispers that there is something out of the ordinary. For many, comparing little Timmy's physical features and development to his twin brother Tommy brings them to ask the question, why? And for some families, at the very latest, even society will be asking questions as to why Jyoti is so different to her friends. A very scientific comparison between her and her ethnic age and sex matched controls. The story I am telling is of families affected by rare diseases across the world, and the journey they are on is to find out what exactly is not quite right, what exactly is causing this out of the ordinary scenario. The journey to a destination, the journey to a diagnosis, often the so-called diagnostic odyssey. What does this odyssey look like? Unlike you and I, when we visit the GP with a particular complaint, he or she may do a test or an X-ray and then prescribe some treatment, and a few days later we'd be 100% again. For families with rare diseases, this journey to a diagnosis becomes a long, painful, and unsatisfying odyssey. An odyssey punctuated with multiple tests, investigations, some painful, some invasive, and many totally unnecessary. These conditions are complex, often multisystemic, and therefore inherently complicated, making the diagnosis everything but straightforward. Research in Europe and America has shown that families consult on average 8 different medical specialists, and still the majority is left without a diagnosis. So the diagnostic odyssey continues. On average, in developed nations, a diagnosis is reached after 6 years. So if the situation is that poor in developed nations, can you imagine what the odyssey looks like in Africa? For most patients in Africa, even in South Africa, the diagnostic odyssey is most often lifelong. Most families will never get a name for their condition. And yes, words like rare make it seem that only few people are affected. We know, however, that collectively rare diseases affect a substantial proportion of the population, and they range in their rareness from those common enough for a general physician to have come across to ultra-rare, where only a handful of patients may be affected globally at any one time. Just because they are rare does not mean that we should not care. Why exactly should everybody care about rare diseases? This is a question I get very often. Here are just a few reasons. Half of affected people are children. More than a third of children with rare diseases will not live to see their 5th birthdays. Almost a third of neonatal ICU deaths are caused by a rare disease. Shocking statistics, but still true. You cannot hope to address the United Nations Sustainable Development Goals without focusing on rare diseases. We cannot make a difference to infant and under 5 mortality without focusing on rare diseases. We are not serving a substantial proportion of our populations if everyone does not dare to think rare. Rare diseases are often called orphan diseases because they have historically been ignored by all: clinicians, researchers, governments, and the pharmaceutical industry. And while yes, they may be individually rare, it is vital to understand that cumulatively there are over 300 million people affected by rare diseases worldwide, and we estimate that almost 4 million South Africans are living with a rare disease. These statistics are often based on estimates, as is the case in our country and the rest of sub-Saharan Africa, because we do not have enough studies to know for sure. My mission is to change this and to use genomics to change the estimates into real numbers, because the real people behind those statistics deserve to be prioritized. Medical geneticists care about rare diseases because the vast majority of them are genetic in origin. So most rare diseases are genetic disorders, with over 6,000, some say 7,000, different rare disorders. It really requires a whole specialty in medicine just to deal with these. Do I get overwhelmed by these statistics? Not at all. On the contrary, knowing this, that the majority is genetic, gives me a lot of hope that we will be able to diagnose the majority. We have the technology and we have the skills. While in the past, medical genetics has been seen as a dysmorphology-based discipline only, our role as medical geneticists now, especially in the last few years, has evolved dramatically and includes being experts in many areas from basic biology through to genomics, through applications and implementation of new technologies, and much, much more. So what is the current situation in Africa like? The honest answer is we really have no idea. Most of the continent, especially sub-Saharan Africa, still has huge gaps when it comes to the care of people living with genetic disorders. Few countries have medical genetic specialists. Even fewer have access to genetic counselors. Even fewer have laboratories set up to do genomics. The infrastructure is missing. While we have been focused on infectious diseases like HIV and TB, the genetics patients have been neglected. What is urgently needed is a skilled workforce to deliver medical and clinical genetics services, laboratory services, especially laboratories equipped to do genetic testing, things like patient registries, and other support structures, including patient support groups, avenues to access care and treatment, and importantly, to be able to do this all in-house, without us having to export samples to the global north and to be victims of helicopter science. We will ride the infectious diseases wave, which has at least brought the technology needed to the continent. For everything else, we need significant buy-in from government and other stakeholders. In South Africa, there are currently 5 centers across the country offering medical genetic services. A total of 15 medical geneticists work across public and private practice. What becomes glaringly obvious is that looking at this map, we see that 6 of the 9 provinces don't have access to medical genetics. There is a lack of political will, and we've been struggling for decades to train geneticists, only for them to have no post once they qualify. And while 6 of the 9 provinces need their expertise, outreach clinics do take place, but they are not nearly sufficient for the needs of the rare disease communities in those provinces. Ideally, I'm advocating for increasing the number of training posts and consultant posts, and the establishment of centers of excellence across the country to cope with the needs of the communities closer to their homes. As a member of BRICS, with Brazil, Russia, India, and China, South Africa is the farthest behind in terms of moving forward with genomics and incorporating genetics and genomics into routine care. Currently, testing in our National Health Laboratory Service still only offers very few tests. Thankfully, we do have access to chromosomal microarray, but these are mainly requested by medical geneticists, as the average pediatrician or physician would not be equipped to order such a test. We have several single gene tests, mostly for the common variants, and still mostly Sanger-based for conditions like achondroplasia, for example, or for founder mutations in our population. What I'm saying is that there has not been an adoption of genomics on a national scale. There's been little support for NGS-based testing, for gene panel testing, never mind exome and genome testing, despite the evidence from elsewhere in the world regarding the benefits of these tools. When I returned to South Africa at the end of 2019, this is the first thing I needed to change. I lead the Rare Disease Genomics in South Africa Research Group, currently an all-female group based at the medical campus of Stellenbosch University, which is on the same campus as Tygerberg Hospital, where I'm also on staff. Our motto is, until every African patient with a rare disease is diagnosed. In different ways, we use genetic and genomic testing to get a diagnosis to as many undiagnosed patients and families as possible. One of my graduate students was Dr. Farida Essagi, a fellow in pediatric neurology. When I arrived back in South Africa and started working at Tygerberg Hospital, I recognized the need for early testing in children with early onset epilepsy. This was a group of patients where fast diagnosis could have a real life impact, and for whom we know there is a huge burden on the healthcare system in the absence of genomics. I wanted to leverage what I knew we would find in this population to gain acceptance for genomics across the hospital. The hospital administration agreed to pay for a gene panel, an epilepsy gene panel comprising 300 genes. Farida then enrolled almost 50 patients and found a diagnosis for almost half of them. For the diagnosed ones, a third had a change in management and the opportunity for precision medicine. This has been phenomenal in the pediatric neurology clinic, such that genetic testing is now becoming second, if not first-line testing. The hospital has agreed to continue paying for panels because they recognize significant cost savings and the prevention of repeat procedures and the benefits associated with definitive diagnoses, which include prognosis, recurrence risk, and accurate genetic counseling. The next step is to widen our array of panels that are covered by the hospital on the one hand, but also to do more in terms of genomics on the research side. We need data in order to secure buy-in from important stakeholders so that genomics can move out of the research realm and into clinical routine care. This is where the Undiagnosed Disease Program in South Africa comes in. I established this program at the end of 2020, so it has been running for almost 18 months now. We have prioritized sick young children in the first leg of the UDP, as we believe that testing early can have the greatest impact. But I also firmly believe that it is never too late for a diagnosis, so our activities have started to include more older patients from this year. Our setting is different to other parts of the world, perhaps, where we don't often have access to trios, so the father is often either completely or logistically absent and not able to be included in the testing. The UDP, therefore, has over 90% singletons, a few duos, and very few trios. 99% of families are non-consanguineous. This program is unique in sub-Saharan Africa, and it is the first step to getting the undiagnosed a diagnosis. Other benefits are gathering data on our population, which is understudied and underrepresented in genomics worldwide. This underrepresentation of African people in genomics is well known and comes in many forms. Whether we look at large population databases like NOMAD, or even clinically relevant databases such as ClinVar, there are very few African samples included. Do a literature search and include terms like genomics in Africa, and most of the papers will be regarding infectious diseases, very little on the rare disease population. Therefore, the first leg of my mission was to have my African patients get the same treatment as their counterparts abroad, and this program is the first step to fulfilling that mission. We know that the diagnosis is not the be-all and end-all, but it is a significant start, without which we cannot ever hope to move to more appropriate and newer therapies. Indeed, the data from the last 18 months of sub-Saharan Africa's only undiagnosed disease program has been exceptionally gratifying. We have performed exome sequencing for over 200 patients, got a diagnosis for more than half of them, and for many of my patients, they are the first and only people on the continent to be diagnosed with their particular condition. There are so many benefits that we can already see from the program, which far outweigh the challenges in establishing and running such a program in our setting. It allows for accurate genetic counseling. We've identified common variants in our subpopulations, which has allowed for cheap local testing to be implemented. A good example of this is the common PGAP3 variant, which underlies Mabry syndrome in our country. We've also been able to show the clinical utility of exome sequencing in our understudied non-consanguineous population. There are first steps to quantifying the health economics of doing genomic testing and implementing it sooner rather than later in the diagnostic journey, and these are all very gratifying results which we will leverage to secure buy-in to be able to expand the UDP in future. Along with the benefits come many challenges, however, some of which are easier to overcome than others. We are privileged to work in an amazing team, but this is not the case for many other centers and institutes across Africa. To get the mission fulfilled, everyone needs to be driving the mission forward. We've made strides in the tip of Africa, but there are so many more challenges that need to be addressed. Due to the COVID-19 pandemic and the recognition of the importance of genomics in that pandemic, we now have a NextSeq machine and 2 NovaSeqs in our institute, in our very building, which are up and ready to be used for whole genome and rapid whole genome sequencing for patients far beyond our borders. We are also training the next generation of clinicians and scientists to take up this mission. Courses such as yours help tremendously in upskilling these young scientists, even from far away. What is still needed in order to truly provide the service to everyone who needs it is more support from the governments in Africa and from funding agencies. We cannot do any of this alone, and including everyone in this diagram in the mission is imperative for it to be successful. As I keep mentioning, this mission is an important one, especially in South Africa, but in Africa in general, because the majority of affected people in Africa remain undiagnosed. Everyone has to not only dare to think rare, but once they have to provide every level of support to prevent the families from remaining on their diagnostic odysseys forever. Most African patients and most African families never learn the name for their conditions. Families face stigma and psychological challenges with no answers, and that is frankly not only unacceptable, but it is unconscionable. The future is really enabled by genomics. It's an exciting time to be a geneticist, both in the clinic and in the laboratory. Increasingly, we're getting access to new tools to interrogate our genomes and to use this information for better treatments and better health. There's an international call to achieve a diagnosis for all people with rare diseases within 1 year after presentation. This is not a dream. It really, truly can become a reality. We're witnessing a paradigm shift where we're moving towards precision medicine and tailor-making treatments to the variant, and we're using this new information we have to develop new therapies. The future is now, and the future can be now in Africa and the rest of the global South. This mission is not a dream, it is attainable with a very few prerequisites. The implementation of genomics in Africa and the rest of the global South is vital. Our populations are all understudied and all underrepresented. This needs to change. With increased access to genomics diagnostics, we're making small changes here. What we need is for more physicians and scientists to dare to think rare so that it doesn't take decades for our patients to be diagnosed. Dare to think rare so that we can end the diagnostic odyssey before it has even begun. Dare to think rare so we can move onwards towards getting our patients the treatments and interventions that they need. Dare to think rare so that all patients with rare diseases can receive the same care their peers do overseas. And for that, we need all of you on this course especially. Remember that behind every data set that you're working with is a real person waiting for an answer. At the end of the course, you'll be equipped to provide those answers. It is a privilege to be part of putting an end to the diagnostic odyssey. I wish everyone lots of success in this amazing course. Thank you very much for the opportunity to speak to you today. I hope that you will join me on this mission to get genomics implemented in whichever country and whichever region you work in, so that we can benefit those who really need our input. Please feel free to contact me, and if you're on Twitter, you can follow the research group there. Thank you very much.
Hope you had a wonderful treat listening to the experts in the inaugural session of the Genomic Variant Analysis and Interpretation course. I would use this opportunity to thank the speakers for sparing their valuable time to enlighten us, and we look forward to having you all for the next 16 weeks for the second edition of the Genomic Variant Analysis and Interpretation course. The course will be administered on this portal, and over the next 16 years, you would have specialized sessions which would educate you and inform you about the various aspects of evidence collection, evidence curation, and evidence interpretation for independent evaluation of the pathogenicity of genetic variations. We sincerely hope that the sessions will be very educative. and we'll be more than happy to hear your feedback on the sessions, and we look forward to the continued engagement even after this course. Thank you very much. Namaste.